Lesson 3D, Gene Expression or Protein Synthesis. Translation. In this step of protein synthesis, the mRNA created in the transcription process will travel to the ribosome and be translated into a protein. Translation occurs in 3 steps as well, using the same terms: 1. Initiation, 2. Elongation, and 3. Termination. Before we look at the mechanics of building a protein, we will introduce to you the 3 chemicals involved in the process. First is the mRNA strand. This is the RNA copy of the DNA gene that was created during transcription, and this holds the recipe for the protein, listing the correct order that amino acids should be joined together to make a properly functioning protein. The next is ribosomal RNA, also known as rRNA. This is actually made of 2 subunits of RNA and is formed in the nucleolus. This is the machine responsible for reading the mRNA and adding the amino acids to the growing polypeptide chain. Ribosomal RNA will read the mRNA nucleotides in stretches of 3 bases called a codon. Finally, there is transfer RNA, or tRNA, which will bring the appropriate amino acid to the growing polypeptide chain, which is being synthesized by the ribosome. Transfer RNA is made of RNA, and on the bottom of the tRNA molecule, 3 bases are exposed, which allows for it to complementary base pair with the mRNA temporarily, while the rRNA synthesizes the amino acids together. With 4 possible bases, A, U, C, and G, and the rRNA reading 3 bases at a time, where repeats are allowed, that means there are a total of 64 different 3 base pair combinations. We call this the genetic code. As you can see, there is a single start code or codon, and there are 3 stop codons. This leaves 61 other triplet codons, but there's only 20 amino acids necessary for protein synthesis. This allows for a redundancy in the code, which all but the amino acid methionine has more than 1 triplet codon coding for that amino acid. In fact, all the others have at least 2, with a few with 4, and serine and leucine have 6 different codons each.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.